Hello friends, let's learn something about how to perform simple staining. Staining is one of the microbiological technique for coloring the microorganism with organic compound dyes. The major components of dyes are chromophore and oxophore. The function of the chromophore is to impart color, whereas the function of the oxophore is to impart salt ion. Depending upon the chemical behavior of the dyes, these are categorized in three groups: acidic, basic, and neutral dye. Acidic dye is one in which charge on ion is negative, or simply it is negativity charge chromophore. Acidic dyes are generally used to stain basic component of cell, that is nucleic acid, example purine acid, India ink, niacin, eosin, etc. Basic dye. Basic dye is one in which the charge carried by the dye ion is positive, that is, it is positively charged chromophore. Basic dye is used to stain acidic components of cell, that is cell wall, cytoplasm, cell blue, safranin, carbonyl, etc. Neutral dye is a complex of acidic and basic dye. It is generally not used in lab, example cosinate of methylene blue. Staining is of three types: simple staining, differential staining, structural staining. The advantage of staining are the cellular arrangement and cells are more clearly visible, whether these are rod, cocci, or spiral in the shape. Difference between cells of different species and within the same species can be demonstrated. Apparatus required are glass slide, inoculating loop, Bunsen burner, and microscope. Chemicals required are suspension, methylene blue or crystalline violet oil for immersion. Hence, the cocci in cluster, rod in chain, and the oval yeast cells are absorbed in simple way by microscope after staining in dye, respectively, with the help of simple staining using basic and acidic dyes. For procedures and the precautions how to take during staining, you can read as shown in the book cover. Thank you.